entitled Autotetraploid Plant Regeneration by Indirect Somatic Embryogenesis from leaf mesophyll photoplast of diploid Gentiana decumbens LF species. This article was written by Karolina Tonizek, Anna Mikula, Elvina Sliwinska, and Jan J. Ryberzynski. Okay, from the introduction of this article describes about the Gentiana decumbens species, which is a triploid species. Gentiana species is a perennial herb and growing on grassland slopes. There are so many types of gentians. So, Gentiana is a source of beta-saccharoidal glucosides and flavonoids, and it possesses high antioxidant potency both in vitro and ex vivo. Gentiana decumbens. So, this species also used in medicine in Mongolia as a treatment for liver disease, and it was in Pakistan. It's also used to prepare as a stomach treatment with stomach disorders. So in modern biotechnology, it's often important tools for mass propagation and genetic improvement of medicine and ornamental plants. So from this article, the aim of this work was to develop a protocol for efficient plant regeneration from protoplast and differentiate green leaf mesophyll cells of Gentiana decumbens. The regenerants were evaluated using both direct and indirect methods to estimate ploidy. So how this experiment works? For your information, this work starts by obtaining seeds of Gentiana decumbens and being sterilized around the surface of the seed by 70% ethanol. After that, we follow the sterilized with 1% of sodium hypochlorite and rinse it 3 times. The seed will be placed on half-strand mulching and scum medium. The plantlets will subculture to a new medium every 3 to 4 months. After that, young leaf will harvest from in vitro ground and Gentiana decumbens. After that is begin the next step, which protoplast isolation. In protoplast isolation, the leaf we take and remove the lower epidermis, and we take the leaf tissue and incubate it in filter sterilized enzyme solution for 3 to 4 hours. This work must in the dark at 26 degrees Celsius. So after that step, the next step is microscopic evaluation of protoplast. This is to observe the protoplast, and is followed by the protoplast culture. After that, in protoplast culture, we must note that to avoid browning of the culture, 2.5 milliliter of the liquid medium was replaced with the same amount of the same fresh medium every week. After that, the important step is color culture. In this color culture and plant regeneration, this work has been used 10 different medium as this table. So next, first step is flow cytometry, followed by chromosome number evaluate, and then we proceed to the size and frequency of stomata, and the last one is statistical analysis. Okay, for the result and the discussion in this article, I would like to discuss about somatic embryogenesis that started in color culture and plant regeneration step that I mentioned before. In this work, the induction of somatic embryogenesis and the development of the first somatic embryos occur after 6 weeks of culture on CPM3 medium, that medium that I mentioned in the table before. So, study have described that plant regeneration from Gentian mesophyll protoplast indicate that the induction of morphogenesis is probably an intrinsic bottleneck in protoplast culture of Gentiana legumes. So this is the picture of the matured somatic embryo before conversion into plantlet. So in contrast, the use of PRM3 medium, which ensure the induction of somatic embryogenesis from callus and cell suspension, and from protoplast of Gentiana cell suspension, it's led to somatic embryo formation from protoplast derived tissue of Gentiana legumes. The highest number of embryos per agarus, which is 2.5, was obtained from tissue culture, previously on CPM3 medium. So it's fewer somatic embryos, which is 1.0 and 0.17, were regenerated from calli formed on CPN4 and CPN2 medium. So which is in CPN3, CPN2, CPN4. CPN3 has a more embryo that obtained from the tissue either than CPN4 and CPN2. So somatic embryogenesis was also induced on other PRM media but with lower efficiency. A repetitive embryogenic response of callus cell was observed on PRM2 medium after proliferation on CPM4 medium with just over 2 embryos that produced per agarus breed. So in conclusion, that we can say that the somatic embryogenesis were obtained from the callus culture and plant regeneration. So that's all that I learned about somatic embryogenesis in this experiment. And so I hope you all understand what I say. That's all. Thank you.